A brain lesion is any abnormality in the tissue caused by disease or trauma. Lesions can result from unintentional causes such as vascular disorders, traumatic brain injuries, and tumors, or can be purposefully created through surgery. Psychologists can learn from both unintentional and intentional lesions, but surgery provides a way of experimentally studying cause and effect relationships. However, because of the ethical problems of cutting into living human brains, most of the well-controlled experiments are based on animal models. Though these studies have provided a wealth of knowledge about the function of specific brain areas and nerve tracts, it's unclear whether the findings generalize well to humans. Additionally, research with non-consenting animals raises its own ethical debate. In addition to lesioning the brain, researchers can learn a lot from electrically stimulating the brain. Once again, this research can take the form of animal experiments or data gained from clinical trials with human patients. One of the uses of electrode stimulation is to use it as a diagnostic tool for identifying important areas of the brain that need to be left intact during surgical procedures. During surgery for the removal of brain tumors, it's important to determine whether the tissue around the tumor is necessary for important functions like movement or speech. By electrically stimulating the tissue while the patient is awake and noting the patient's response, the surgeon can make a safer assessment and perform a surgery with less risk of side effects. Brain stimulation can also be used therapeutically. Researchers are examining the use of deep brain stimulation, electrodes implanted into structures deep within the brain, to treat various conditions. The electrodes are connected to a rechargeable battery pack and stimulator device that's implanted elsewhere on the body, like around the collarbone. Research also suggests that deep brain stimulation may be helpful for severe depression that has not improved after more traditional approaches such as antidepressant medication. One area of the brain that has shown promise is Brodmann area 25. There are a number of innovative ideas related to brain stimulation. Researchers have been able to remotely control cockroaches using implanted electrodes and hope to add small cameras so that these cyborg bugs can be used for search and rescue efforts. One of the most amazing new brain stimulation methods is a technique called optogenetics. Green algae has photosensitive receptors that allow the organism to respond to light. Scientists identified the gene that allowed the algae to produce these specialized receptors and developed viruses that could deliver these genes to the neurons within the brains of mice. Sure enough, these neurons produce the light-sensitive receptors. Researchers then inserted small fiber optic cables into the brain and could selectively activate these neurons on and off as needed by turning on light. Electrodes inserted into an animal's brain typically stimulate hundreds of thousands of cells. Using lesions or drugs also hit circuits like a hammer. Optogenetics could act like a fine scalpel, turning particular neurons in a circuit on or off within milliseconds. If we can record brain activity based on measuring magnetic fields that are produced, can we influence brain activity by applying magnetic fields? This is the idea behind transcranial magnetic stimulation, or TMS. Weak electric currents are induced using a rapidly changing magnetic field, thereby activating or inhibiting areas of the brain with minimal discomfort. These magnetic pulses also stimulate muscles and skin in the scalp and cause a moderate tapping sensation to be felt in the scalp under the coil. The repetitive pulses, the R in rTMS, stimulate a small area on the surface of the brain about the size of a quarter. Researchers are continuing to explore the use of transcranial magnetic stimulation for conditions like depression. Clinics for treating depression using TMS already exist, but there is still a lot that's unknown about this process. I'll end by referencing a study conducted in 2013 that attempted to remotely control another person's brain. One researcher was hooked up to an EEG machine that read his brain activity while he played a simple video game. The other researcher, located in a room on the other side of the building, sat in front of the same game. When the first researcher pressed the fire button, the brain signal was picked up by the EEG, wirelessly transmitted to a transcranial magnetic stimulator placed over the motor cortex of the second researcher, and stimulated the second researcher's finger to press the fire button. It was a very simple action, and it's unclear whether complex behaviors could be manipulated in this manner, but one has to wonder what the future holds.